Greeting friends, in this video we are going to talk about the fates of pyruvate. Look at this man. This is confused now. He wants to know what to do with pyruvate, that is the end product of glycolysis. He wants to know after glycolysis what we are going to do with this pyruvate, where it is going to enter, or what is the further products of pyruvate after its metabolism. Okay, so for that we are going to answer few questions before we get into the fates of pyruvate. The first one is the what about NADH? Remember, what did we produce in glycolysis? That is the NADH after we oxidized the glyceraldehyde 3-phosphate. We discussed that this needs to be further reoxidized into NAD positive so that glycolysis can continue. Okay, we also discussed that it can be continued if it donates its electrons to the electron transport chain, where the ultimate receiver is the O2. It can be possible. It is possible, okay? But what would happen if there is no oxygen? There is no oxygen, then, but we still want to continue our glycolytic pathway, but there is no oxygen. If in the absence of oxygen this is not converted into NAD positive, glycolysis would stop. So for the continuation of glycolytic pathway, even in the absence of oxygen, we have developed some strategies, or you can say other pathways, through which we can convert this NADH to NAD positive to continue the glycolysis. That is what we call the fates of pyruvate. Very well known fate of pyruvate is, we know that it would be going to the TCA cycle, but there are others. The fates of these depends, we can say, depends on the availability of O2, okay, that is oxygen, or absence of oxygen. That is what we are going to discuss. The fates, their types you can see on the screen. So now we are going to discuss the first fate of this pyruvate, that is the lactic acid fermentation, or we can say that conversion of pyruvate into lactate, or simply the reduction of pyruvate into lactate. This is the first fate of, you can say, the pyruvate, where we are going to convert our pyruvate into lactate. So what is the structure of pyruvate? This is 3-carbon molecule, okay, we know that. This is the keto group present here, and this is our pyruvate molecule, pyruvate. This is the reversible reaction, okay. So this pyruvate is going to be converted into lactate. No difference, only hydroxyl group is on the 2nd carbon, brought about by the enzyme. First I draw this structure. So the enzyme doing all this reaction is lactate dehydrogenase. Lactate dehydrogenase is bringing about a change, that is the conversion of pyruvate to lactate. Okay, so we can say that this is the change. Now the next question is, where is occurring? We know the reaction, where this is occurring? Because you can guess that it must be occurring somewhere where there is no oxygen. So the muscle cells, when we do exercise, strenuous exercise, or you can say vigorous exercise, then there is a shortage of oxygen, and to continue to provide the energy, we use this, you can say, convert this pyruvate to lactate, because lactate is converted into this lactate is produced, you can say, when we do strenuous exercise. Okay, so this is okay. But about RBCs, there is also RBCs where this reaction is occurring. Why RBCs? Because RBCs, if we hear this name RBCs, we must say they are containing oxygen. They are transporting oxygen. Blood is responsible for transportation of the oxygen. So why we have included RBCs for this reaction? Because it lacks mitochondria, and we know the site for TCA cycle is mitochondria. Even if it is containing oxygen, then it is not showing this reaction because it lacks mitochondria, and the site for the TCA cycle is mitochondria. So we are not converting, or we are not going to enter this pyruvate into the TCA cycle, or when we convert this pyruvate into acetyl CoA, so it is not done. So the next question is, we can say, any change of NAD positive or NADH. This is interesting. I am going to draw a figure here before we discuss this. How did we start with glycolysis? Glycolysis converted glucose into glyceraldehyde 3-phosphate. Okay, glycerol, two molecules, we can say glyceraldehyde 3-phosphate. This is converted into, you can say, pyruvate. How many molecules? 2 molecules. Pyruvate is getting converted into lactate. How many molecules? Again 2. Okay, so this is, how did we oxidize glyceraldehyde 3-phosphate? With the help of NAD positive. Again 2. To what it is converted to? To NADH, and it is going back. So what we are seeing here is that 2 NAD positives we used to oxidize glyceraldehyde 3-phosphate, so that with the subsequent glycolytic reactions it can be converted into 2-pyruvate. 2 NADs we used. How many we converted to NADH? 2 NADH. And 2 NADH are getting converted into 2 NADs again with the help of the enzyme lactate dehydrogenase. So the question here is that, is there any change of these two entities, these two substances, whatever you say, molecules? But we can say there is no net change. That is we wanted to know. So no net change when we convert this NADH to NADH, or when the glycolytic pathway convert NAD positive to NADH. The next question is, what about lactate? We produced this lactate. What we are going to do with this lactate? This lactate would go into the bloodstream. Okay, from where? From this bloodstream it is going to where? To liver, where it would convert into glucose, and this cycle is known as Cori cycle. We are not going to discuss Cori cycle here. There would be a separate video on this. Okay, so this is about fates of pyruvate. This is only the first fate. In the next videos we are going to discuss the second phase. Thank you.